Okay, so we will continue chapter 5. This is part 2 of chapter 5. Okay, so last time we talked about the functions of cell membrane. And now we'll continue with the cell membrane structure. Just remember that the main framework for the cell membrane, okay, is made of phospholipid molecules. And there are two layers of phospholipids that make the cell membrane. Therefore, it's called the phospholipid bilayers. In addition to the phospholipid bilayers, cell membranes also have many proteins. Okay, and there are two types of proteins. First, based on structure, how it looks like, where it is located. There are two types of proteins on the cell membrane. The first one is called the integral protein. Integral protein, which is the protein that's crossing the cell membranes. Okay, like this one over here. So this is example of integral proteins. The second one is called the peripheral protein. Peripheral means only in one side of the cell membrane. Example is this one, okay, maybe only on the inside or maybe only on the surface of the cell membrane. So these are the peripheral protein. Now, based on the functions, okay, there are 6 types of protein on the cell membrane. The first one is called the carrier. This is the one that carries something to enter or to exit the cell. Mostly the carrier or transporters are integral proteins. The second one is called the channel proteins or ion channel. Okay? And again, mostly these channel proteins or ion channels are integral proteins too. The next function of proteins is for a receptor. This is the one that receives hormone, for example. Mostly peripheral protein. And then the next function of proteins on the cell membrane is as a marker. So markers is the cell that make the cell specific for a specific person. That's the reason why if we get the graft, for example, skin graft, if it is coming from other people's, usually there is a rejection. Because what? Because their cell have different marker compared to our cell. The next one is a function as a linker, which is the one that link one molecules to another molecules. And the last function of protein on the cell membrane is to speed the reaction as a catalyst for the reaction. It is known as the enzyme function. So these are the proteins. In addition of these phospholipid bilayers and the proteins, the cell membrane also contains cholesterol. And cholesterol, because they are lipids, they will be located in the tail. Okay, so around the tail of the phospholipid molecules. Okay, so the functions of this cholesterol is for strengthening the cell membrane, make the cell membrane become stronger. And again, also there are some other molecules on the cell membranes like glycoproteins and glycolipids. Mostly they are on the surface of the cell membrane. So these are the cell membrane structure. Now we go to the next one, which is the transport across the cell membrane. Remember, one function of cell membrane is to control, to manage the movement of molecules in or out. The way they manage it by what we call the selective, selective permeability. It means that the cell membrane able to select, to control which molecules enter, which molecule exit from the cell. There are two main types of transport across the cell membrane. The first one is called the passive transport, and the second one is called the active transport. Okay, passive transport means this transport, this movement of molecule does not require ATP, does not require energy. It's moved automatically by itself. Usually, that movement is called diffusion. Okay, so movement is called the diffusion, which is movement of molecule, okay, from higher concentration area to lower. Okay, so from higher to lower concentrations area. Therefore, they do not require the energy. There are several types of passive transport. The first one is called the simple diffusion. The second one is called the facilitated diffusion. And the last one is called osmosis. We will discuss this later, okay? And then for the active transport, okay, from the name, it is active. It means that it's require ATP, require energy. There are several types. The first type is called the primary active transport. And we have a lot of this primary active transport in our cell membrane. They are called as a sodium potassium pumps. So this is the pumps. Okay, remember, if we have the pump, it means that we have energy to pump something, whether in or out from the cell. The second one is called the secondary active transport. For this secondary active transport, there are several types, whether uniporter, symporter, or antiporter. Again, we will discuss this later. And the next one of the active transport is called the bulk transport. Bulk transport is the active transport that use physical. Use physical to transfer bulk or many molecules at the same time. This is just like, you know, a vehicle, like bus, for example, that carry many students, for example, or many people in one time. So this is what we call the bulk transport. And there are two types of bulk transport. If it is enter into the cell, it's called the endo. Endo means enter. So this is endocytosis, enter the cell. The second one is called exit from the cell. Therefore, it's called the exocytosis. This is exit from the cell. So the bus or the vehicle, the physical, exits the cell. For the endocytosis, there are three types. Phago. Phago means eat. So this is cell eater, phagocytosis. Pino. Pino means drink. So this is like a drinking. And then there is another one, is called the receptor-mediated endocytosis. We will see this one later. Okay, now we start with the passive transport. Okay, passive transport, which is the transport does not require the energy or ATP. Again, the transport mostly is called as a diffusion. Movement of molecule from higher concentration area to the lower concentration area. The first type is called a simple diffusion. This is for small hydrophobic molecule like oxygens and carbon dioxide. If we, for example, making the cell membrane like this, Remember, it has 2 layers of phospholipid bilayer. Okay, and then let's say we have a blood vessel over here. Okay, so blood vessel carry a lot of nutrient. Okay, okay, such as glucose and oxygen. So this oxygen will be dumped into the area between the cell and the blood vessel. Okay, so there will be a lot of oxygen over here. So this is higher concentration area. And oxygen will enter into the cell, okay, easily. This is what we call the simple diffusion. Inside the cell, okay, the oxygen will be used to convert the glucose into ATP, okay, plus carbon dioxide plus water. And it is happening in an organelle is called the mitochondria. So if you see here that the oxygen enter from higher concentrations, enter into the cell because the cell have lower oxygen concentration. And this is what we call the simple diffusion. If you see over here, the results, okay, because oxygen used to convert the glucose into ATP plus, there are a lot of waste product, carbon dioxide. This carbon dioxide will be more or higher inside the cell. Therefore, carbon dioxide will out from the cell easily. Okay, and it will go to the blood vessel, and this will be carried by the blood vessel as a waste material. It will be carried into our lungs to be exhaled later in the lungs. And the carbon dioxide also use the simple diffusion type of transport. Now look at the glucoses over here. Okay, blood vessel carry a lot of glucoses. Okay, let's say glucose is over here. It's going to be higher concentration outside the cell. And because glucoses are too big, very big, they cannot go through this phospholipid bilayer. These glucoses require what we call the carrier or transporters. So this transporter will be used by the glucoses to enter into the cell. This is still diffusion because from higher to lower concentration. However, in this movement, glucoses need facilitation, need help from the carrier protein. Okay, therefore, the movement of glucoses is called the facilitated diffusion. And because it's using the carrier protein, okay, so this one, this glucose move with facilitations with carrier proteins to enter into the cell. Now we also have, okay, our blood carry a lot of ions, okay, let's say sodium, for example, a lot of sodium outside the cell. And then we need sodium inside the cell. So the sodium will enter into the cell. However, because the phospholipid bilayers are lipids, which is hydrophobic molecules, it will not take the ions. Okay, so this ion is hydrophilic. This is polar molecule. Okay, so hydrophilic molecule. So hydrophilic molecule will not cross the hydrophobic layer. Therefore, this sodium will use another type of protein. It's called a protein channel. Protein channel or ion channel. Okay? So the movement is still from higher concentration to lower concentration, which is diffusion. However, this require channel proteins or ion channel. Therefore, this ion also move by facilitated diffusion. Okay. Now, osmosis. How about osmosis? Osmosis is special movement for water. Remember, we have water here inside and we have water outside. Now, where the water will go? That's the question. the water will always go from a lower solute concentration to higher solute concentration. Like, for example, if we have too many sodium outside, let's say sodium chloride outside over here, okay? So the water will move out in order to try to dissolve this, to make it balanced. So a lot of water will go out because so many sodium chloride outside. This water movement, they usually use a channel. Okay, this is what we call the water channel. Or aqua porine. Okay, to move from lower solute to higher, okay, higher solute concentration, using the aquaporin. And this is what we call the osmosis. Okay, so there are again three types of passive transport: simple diffusion like oxygen and carbon dioxide, and then facilitated diffusion for bigger molecules like glucose, amino acid, and then also facilitated diffusion for ions, sodium ions, chloride ion, potassium ion. A lot of ions require this ion channel or protein channel. And the last one is the osmosis, movement of water. Now we're gonna see the osmosis and tonicity. Okay, how this water actually move, okay, from one place, one area to another area. Remember, osmosis is the movement of water, okay, movement of water, okay, from low solute concentration to higher solute concentration. Okay, now let's say we're going to use red blood cell, for example. Let's say we have 3 containers, okay, 3 containers like this. Okay, we put solution in it. Okay, and then we put, let's say, red blood cell. Red blood cell. Over here, we put red blood cell. We put red blood cell. Okay, so red blood cell, okay, actually has sodium chloride in it. It has around 0.9% sodium chloride. Okay, so 0.9% sodium chloride in red blood cell. 0.9% sodium chloride in red blood cell. Okay, the first solution over here, okay, we do not put any salt. So this is 0% salt in this solution. Because the solution has less concentration of sodium chloride, the solution is called the hypotonic solution. The solution that has less solute concentration. On the second container, we put 0.9% sodium chloride. Okay, so we dissolve sodium chloride, it reaches 0.9. It means that this solution has the same number, the same concentration of sodium chloride with the red blood cell. Therefore, this solution we call as an iso. Iso means the same. Okay, isotonic solution. Let's say on the other one over here, we put 10% sodium chloride. Okay, it's very salty. Okay, so this is salty solution. Okay, because this has higher solute concentration, this solution is called a hypertonic solution. Okay, now look what happened when the water will go. Remember, in the red blood cell, there is water. Okay, and this is water. Inside water, outside water. Inside water, outside water. Okay, now where the water go? Remember, the water go from low solute concentration to higher solute concentration. So which one is low? Outside is only 0 and inside is 0.9. So what happened? The water will enter into the cell, into the red blood cell, right? And then red blood cell become bigger, bigger, okay? And then finally it will explode. It will broken down. And this process is called a hemolysis. Okay, hemolysis means the red blood cell will be broken down, will be exploding. Okay, so this is the what happened if we put a cell in the hypotonic solution. The cell will be broken down, will be lysed or hemolysis. Now look at this one on the second one in the isotonic solution. Because the concentration is the same, then the water will enter, will exit, enter, exit at the same way. Okay. So what happened with the cell? It will stay fresh. Stay fresh. Okay. It's nothing happening to the cell. The cell stay fresh. This is the way when we put a cell to preserve the cell, we put the cell in the isotonic solution. Sometimes this solution is called as a buffer solution, the solution that keep the cell fresh. Now look what happened with the hypertonic solution. Remember, the water go or move from low to higher. Inside is lower, 0.9. Outside is higher, 10. So the water will out from the cell. Okay, what happened with the cell? The cell becomes smaller, become shrinking. Okay, so this process is called the crenation. When the cell is become smaller, shrinking. Okay, so this is what happened if we put the cell in the hypertonic solution. The cell will be smaller, will be shrinking. If we put in the hypotonic solution, the cell will be bigger, and one time it's become explode, hemolysis. If we put in the isotonic solution, there is nothing happen with the cell. The cell keep fresh or stay fresh. Okay, now we go to the second type of transport. This will be second, okay? Second type of transport, which is active transport, okay? We will see the primary and secondary active transport. Let's say this is the cell. Okay, this is the cell. Okay, in the cell we have a special pump. Okay, it's called the sodium potassium pump. Okay, that will pump the sodium ion and potassium ion in or out. This is actually what's happening. We have more potassium inside the cell. So there are more potassium inside the cell. And there are more sodium outside the cell. Okay? And then remember, the sodium has the channel. It's called the sodium channel. Remember, facilitated diffusion? Okay, this is sodium channel. Okay? So sodium will enter into the cell. Every time sodium enter into the cell, there will be 3 sodium enter into the cell. Okay, and the potassium also have potassium channel. Okay, potassium channel. This is potassium channel. Okay, remember diffusion, it will go from higher concentration to lower concentration. So potassium actually will exit from the cell, but only 2 potassium exit from the cell. Now forget about the potassium or the sodium. Just remember the chart. So if you see over here, there are 3 plus enter, okay, and there are 2 plus exit. What does that mean? It means that there will be always extra 1 plus, okay, 1 chart. It is not good. the cell will die, okay, if there is no balance. Therefore, the cell have this pump over here to make this balance. So this pump is actually will pump in, remember, the active transport is movement of molecules using energy from lower to higher concentration. So it's gonna be pumped into potassium, okay, because there are more potassium inside the cell, they need pump. At the same time, okay, the pump also will pump 3 sodium out. Yeah, you see this one? So you said 3 balance this, 2 balance this one. Okay, so 2, 2, 3, 3. So therefore, this pump over here, sodium potassium pump, is the pump that is really needed by our cell. Therefore, it's called as the primary active transport. We have to always do this pumping, otherwise the cell will have problem, will die. Okay, so this is the primary active transport. The second one is called the secondary active transport. Okay, this is the movement of molecules using the also carrier proteins. Okay, from lower to higher. Okay, remember lower to higher. And if it is only one movement like this, it's called the uniporter. If it is two movement, okay, using this pump, okay, if it is moving at the same direction, so let's say this is A, and then A will carry B at the same movement, let's say enter into the cell. These two movement is called symporter, movement of two molecules to the same direction. Now, but the last one. So let's say we have another pump over here. Okay, when the A enter, it actually will give the energy for B out from the cell. So this one, what we call the antiporter, movement of molecule to the opposite direction using the secondary energy, okay, coming from the first molecules over here. So this is what we call the secondary active transport. And then the third type of active transport is the bulk transport, okay, which is using the vesicles or vehicle to transport many molecules at the same time. This is what happened, okay? There are several types. If it is entered to the cell, it's called the endocytosis. We're going to see the endocytosis first, like this. So let's say this is a cell, okay? This is a cell, okay? And there is a molecule that's carried by vesicle, okay? Carried by this vesicle. So this cell able to create a mouth structure, just like a mouth, okay, mouth, to eat this, okay, and then this will be eaten and then go inside the cell, okay, inside the cell, maybe it will be released like that. So if it is happening like this, the movement of bulk transport or vesicle enter into the cell by making a mouth structure, like eating, okay, this movement is called the phagocytosis. Phago means eat, okay. The second one, the pinocytosis, is just like this, okay, the same. Let's say the cell, okay, and then there is another vesicle over here. It's just kind of fused together and then finally enter into the cell without making the mouth structure. It's just kind of drinking. So this is what we call the pinocytosis. Okay, this one is phagocytosis. Okay, so this is pinocytosis. And then the next one is called receptor-mediated endocytosis. This is usually, okay, from, let's say this is a cell, okay, and this is inside. So this vesicle is just, there is a receptor on this cell, receive this vesicle, and then they enter into the cell. But this vesicle actually not stay in the cell, but it just go through the cell and then finally release under the cell. Okay, so it's actually crossing into the cell. It's not used by the cell itself. Okay? It will be used by the next area, maybe below this line of cell. So this is what we call the mediated endocytosis. Okay. Now the last one, exocytosis, is very simple. So if it is the cell, okay, and then let's say, remember Golgi apparatus produce hormone, that hormone carried by the vesicle, and then it's going to be fused with the cell membrane, and finally will release from the cell, go to their destination. This is what we call the exocytosis. Okay, so these are the type of transport across the cell membrane. So I think we finished chapter 5. You should be able to fill out your assignment too, especially chapter 5. Finish them all. I'll see you again later. Goodbye.